Hey guys, Mr. P. In this video, we're going to focus on water movement with and without cell walls. So there are cells that have cell walls and there are cells that lack cell walls. And we're going to focus on how the cell wall impacts water movement into and out of a cell. This is the second video of D2.3, and we're going to focus on objectives 5, 6, and 7. Objective 5 says water movement without cell walls, and we're going to include swelling and bursting in a hypotonic medium and shrinking and crenation in a hypertonic medium. We're also going to include the need for removal of water by contractile vacuoles in freshwater unicellular organisms, specifically paramecium, and the need to maintain isotonic tissue fluid in multicellular organisms to prevent harmful changes. Objective 6 focuses on the effects of water movement on cells with a cell wall, and again we're going to focus on hypotonic and hypertonic medium. But when a cell has a cell wall, the development of turgor pressure helps to withstand too much change. And then Objective 7 talks about the medical applications of isotonic solutions and the importance of isotonicity in medical applications. And so we're going to include a discussion of intravenous fluids, so IVs, given as part of a medical treatment, and bathing organs ready for transplantation as examples. So the first thing we need to talk about is what happens to cells without a cell wall when they are placed into a solution that contains various solute concentrations. Again, in the last video, we talked about what isotonic, hypertonic, and hypotonic means. Remember, hypertonic refers to a solution that has higher solute concentrations, and therefore water, because it's higher concentrated inside the cell, will move out. Hypotonic solution refers to a solution that has lower solute concentrations, and so because the cell has lower water concentration than the environment, or higher solute concentration, the water is going to flow in, causing the cell to expand, swell, and eventually burst or lyse. And isotonic refers to a solution that has the same solute concentration, and so because the solution outside of a cell and inside of the cell has the same solute concentration, that dynamic equilibrium is going to be reached and water will move into and out of a cell at the same rate. So cells that lack a cell wall would include all of your animal cells, and that includes human cells. And so that's why in this particular picture we are focusing on red blood cells, but this happens to all of your tissue cells. None of the cells in our body or in any animal is going to contain a cell wall. That's one of the distinguishing characteristics of an animal cell versus a plant cell. Both of those cell types are eukaryotic, and so they contain a nucleus. All of this has been discussed in previous videos, but one of the main differences is that they lack a cell wall or they contain a cell wall. So cells that lack a cell wall are especially vulnerable to the effects of osmosis and will immediately result in the change of physical size and shape. Again, you can see that these cells all started as the same shape and size. They're all red blood cells. They're very uniform, but the size and shape of the cell immediately changes once placed into a solution with varying solute concentrations. Cells placed in a hypertonic solution will shrink, resulting in a term called crenation, which is extreme shrinkage where the cell becomes crinkled, which is why all of these cells look like they're shriveled up. Obviously, structure dictates function, and we talk about that specific statement a lot throughout the IB syllabus or biology in general. And so if these red blood cells are designed to work correctly when shaped like this, you can obviously tell that when they shrink or shrivel up, they're probably going to lose their function, and in fact they do. Cells that are placed in a hypotonic solution, which is a solution that contains less solute than in the cell, are going to actually swell and potentially burst. Obviously, that too changes their shape, which changes their function, and it's not hard to believe that cells, if they rupture, are going to be completely rendered useless and will die. Water moving in, that's because there is a higher water concentration outside of the cell than there is inside. Water moves high water concentration to low water concentration, or you can think of it as water moves from low solute concentration to high solute concentration. So if water is high or higher than it is inside, the solute concentration by default will be lower than the solution inside the cell, which in this case, if water is moving in, will have a higher solute concentration and therefore a lower water concentration. What are they trying to reach? Equilibrium or isotonicity, which is that dynamic equilibrium where water moves in and out at the same rate. this would be one water molecule moving in to every one water molecule moving out. It is really important to identify the fact that water, or the net movement of water, never actually stops. It will continue to flow in and out. So, transitioning to the contractile vacuole of unicellular organisms. So this is a paramecium, and paramecium is a single-celled eukaryote, meaning it is a protist that lives completely unicellularly. This is one cell, and you can see that the contractile vacuole, which is that kind of opening and closing circle in two different places, or right in the middle, is the contractile vacuole, and the purpose of that contractile vacuole is to fill with water and to expel that water. If we were to zoom in and look at that contractile vacuole in close detail, you can see that it actually fills with water and then will expel that water as a way of regulating the osmolarity or the isotonicity of that particular organism within its environment. And we know from that previous slide that if a cell takes in too much water, it's going to swell and eventually burst, and that's going to be bad for the cell. And so if this single-celled organism is in a hypotonic environment, it's going to absorb water, and in order to maintain health and maintain viability in its environment, it has to have a way of excreting that water and getting it out before it actually swells too much. Pretty cool adaptation. Okay, so that is a unicellular organism and its ability to maintain isotonicity, or its ability to withstand water movement without a cell wall. But now let's talk about actual tissue cells within a multicellular organism. So we have very similar structures to unicellular organisms, but we're a multicellular organism, and so we maintain our tissues of a variety of single cells all put together into a very uniform tissue. And so we're looking, in this case, at skin. You can see that when the cells are hydrated, there is a really strong protective barrier from the outside of the environment, and it actually keeps a lot of the unwanted pathogens, both bacteria and viruses, out. And so well-hydrated cells perform their functions correctly, and the function of our skin is to maintain a barrier between the inside of our body and the outside of our body. When cells lose water or become dehydrated, they actually shrink and begin to malfunction. And you can see that these cells, when they shrink, actually pull away from the cells that they're adjacent to, and that creates openings, small kind of micro tears, that allows these pathogens that are supposed to be kept out, they are able to get in, and that's how the barrier, the skin, starts to malfunction, and that leads to disease, and it can lead to illness. And that is bad for us as a multicellular organism, because our skin, the barrier that keeps us safe and healthy from the pathogens in our environment, once it starts working incorrectly. It's all related to water movement. We lack cell walls, and so we have to maintain a certain level of hydration, not just in our skin, but in all cell types, in order for our tissues to work correctly, which leads to our organs working correctly. What happens when water moves through a cell with a cell wall? These cells work differently than cells that lack a cell wall because there is a cell wall present. Incoming water in a hypotonic environment, again, hypotonic leads to water moving in, causes the expanding vacuole in a plant cell, presses the membrane against the rigid cell wall. And so as water moves into these cells from hypotonic to hypertonic, it causes this vacuole to swell with water, because that's the organelle that is in charge of storing all of this water. And as this central vacuole fills up, it's going to push all of these surrounding cytoplasm and organelles more forcibly against the membrane, which forces the membrane against this thick, rigid cell wall, and it actually creates a pressure inside of the cell that we call turgor pressure. A plant cell that is well hydrated is said to have high turgor pressure and is said to be turgid. When water is lost from a plant, meaning if the plant is dehydrated and wilting, the cell membrane pulls back or isn't forced so forcibly against the cell wall because of the lack of water in the central vacuole, and it causes the cell wall to kind of cave in on itself. There's no pressure from within pushing out, and so we call that either flaccid or plasmolyzed, depending on how much water is lost. It results in the plant cell shrinking. It results in the plant wilting, which means it doesn't have as high of turgor pressure. the plant starts to wilt, and that's because the vacuoles within all of those plant cells are shrinking, which causes the cells of the plant to shrink. And as the plant cells shrink, they aren't as rigid, and it causes the plant to be really flaccid. So what are some medical applications of isotonic solutions? Again, it is important. Hopefully you identify the importance of isotonicity. But within a medical application, we have both ICF and ECF. ICF is intracellular fluid, so that is the fluid inside of a cell. We also have ECF, which is extracellular fluid, which is the fluid outside of a cell. And so there is a membrane or a boundary that is formed between this ICF and ECF. And since both ICF and ECF are solutions that are water-based and aqueous, it's important to appreciate that there will be water movement through that membrane from either the ICF to ECF or ECF to ICF. And so you have to maintain isotonicity between the fluid inside of a cell and outside of the cell in order to maintain proper hydration levels within your cells. If you have proper hydration levels within your cells, you're going to have proper hydration levels within your tissues, which means your organs are going to function correctly, and it's a really important deal. So if the ECF, which is the extracellular fluid, has a greater solute concentration than the ICF, meaning it is more highly concentrated, the solute, outside of a cell than it is inside the cell, water will leave the body, resulting in the shrinking of cells and possible fulcrination. So if we place our tissue cells into a solution, or we have a solution outside of our cells in our body that has a higher solute concentration than inside the cells, our cells will actually begin to shrivel up and shrink and possibly even fully crenate. If, however, the ECF has a lower solute concentration than the ICF, meaning inside of our cells has a higher solute concentration than outside, then water is going to enter the cell, possibly resulting in swelling and even bursting if there is enough water movement. We call that hemolysis, meaning cells rupturing. It is important to ensure the maintenance of the shape of our cells, that the ICF and ECF has to be in balance, meaning there has to be an isotonicity to our ICF and ECF. If that is the case, there will be a dynamic equilibrium reached, which means water again will move in and out of every single one of our cells correctly, equally, and it allows for no net movement of water, even though the water moves equally in both directions. A few more medical applications is through the idea of IV. So we place a lot of things into our body via IV, so intravenous fluids. Food, blood products, and medications can be administered through a line, which we call a catheter, when placed in a peripheral vein. And that is one way that we get a lot of fluids into the body in a relatively quick and efficient manner. And in times of organ transplant, there are a lot of steps involved in the preparation of that organ. But it is important that you understand and appreciate that an organ, when kind of preserved or prepped for organ transplant, is actually placed or bathed in an isotonic solution, because you want to ensure that water is moving in and out of that organ to ensure that it isn't swelling, that it isn't leading to plasmolysis or hemolysis, it isn't swelling too much and potentially rupturing the cells that make up that tissue. And so if the organ is immersed in an isotonic solution during the period before it is transplanted, it will avoid cell damage and therefore work correctly once the organ is placed into the recipient post-surgery. That's it for this particular video. If you learned something, give it a thumbs up, subscribe to the channel. We'll see ya.